Thank you. So my name's Elizabeth Eisenhower, and I'm going to be talking about sampling design for animal movement. And specifically, I'm talking about a sampling design that we're calling a lattice and random intermediate point sampling design. And for short, we like to call it LARI. So you can see in the background of this main slide, there are some ants that are kind of moving around. And so these are the ants that are generating the data that I'll be talking about for the most part today. So Hughes Lab at Penn State collected this data. It's very high resolution ant movement data. And they collected it over 4 hours. So basically there's a little man-made nest and they put a GoPro on top of it, recorded 4 hours of data, brought that video back to the lab. And then they had students who sat at computers and clicked on every single ant at every 1 second interval. So they ended up with this amazing data set. So here's all the data in one plot. You can see the boundaries of the nest, and then each individual ant is colored with a different color. However, this took a really long time, and they did not want to have to do this again. So we were approached by these researchers who were wondering, next time if we collect lower resolution data, if we collect less data, how can we minimize the loss of information about movement behavior? So obviously, if we have less data, we might lose some information, but how can we minimize that? And this question is relevant to a lot of researchers. Basically, anyone collecting animal movement data, because there's always some type of restriction when you have, battery life of tracking devices or anything like that. There's usually some restriction to how much data you can collect. So we hypothesized that maybe we could sample differently from what they had done in the ant study and what a lot of people do, which is just kind of regular sampling. So for example, every 5 seconds. Is there another way that we could sample where we could gain more information? And so this lattice and random intermediate point design was actually motivated by the lattice plus close pairs sampling design in geostatistics. And the thought here is, so instead of every 5 seconds, you have one sample every 10 seconds, and then in each 10-second interval, you have one random time point that you're looking at. Okay, where is the animal at this time point? So you end up with the same amount of data, but the sampling design is a little bit different. And the idea is that if you have a range of time intervals, hopefully you'd be able to capture movement behavior at different scales. So for the ant data set, we wanted to compare the full data, which is of course every 1 second, to every 3 seconds, every 5 seconds, and then the LARI 10 seconds. So the every 3 second is just kind of an in-between to see how we're doing. And the one thing to note here is that these last two sampling designs, these are going to be subsamples of the full data, they both have the same number of data points. So the only difference between these two is the sampling design. And so when we compare these two, we're actually directly comparing the sampling design for this data set. So how do we do this? So the model that we are using is a stochastic differential equation model. And so you start with the data, say you have the full data set, then you have this x sub t, it's just the x and y position of the ant at time t. And then the framework looks like this. So it's similar to maybe other things that you've seen at this conference and a pretty common framework. The two equations are, the first is just saying that the time derivative of position is velocity, and the second is saying the time derivative of velocity is acceleration. And then we're able to model acceleration using a sum of these different forces. So beta here is actually controlling the autocorrelation. And then we have mu of x sub t, which is a mean drift term. And then c of x sub t here is the magnitude of the stochastic term. It controls the magnitude of the stochasticity. And w sub t is just two-dimensional Brownian motion. So the thing to note is that that mean drift term and the magnitude of the stochastic term both are functions of motility and potential surfaces. So it's kind of hard to see from this, what that actually means. So I'm going to try to give a more intuitive explanation. So the potential surface, so we can imagine one example of a potential surface would be, say, a bowl, and you kind of have the center of the bowl being at the bottom at the origin in this case. And then you can imagine that an individual would move around similar to how a ball might roll around in this bowl. So it would look something like this, where they're kind of moving around, staying close to that center origin or that attractor in the center, and sometimes venturing off, but always coming back to that location. So that's one example of a potential surface and how that would work. And then the motility surface can be best illustrated by looking at the actual ant data. So you can see here that the outline of the nest is shown, and then the ants are different colors and shapes. So you can see the ants actually kind of spend most of their time in the two outer chambers, and they move really slowly there. And then when they go through the center, they move much more quickly. So we can use motility surfaces to understand this by saying, okay, in the center where they're going to move really quickly, that's high motility. And then in the two outer chambers, that's a low motility. So the surface will have lower motility in those areas where they move really slowly. And so, of course, we don't observe animal movement in continuous time. So we have to do numerical approximations to approximate these derivatives. So I'm switching notation a little bit from t to tau. So t was time, and now tau is the ordered observation. So x sub tau is just the position of that ordered observation tau. And then h sub tau is the time between observation tau and observation tau plus 1. So I changed from t to tau so that we can have different amounts of time in between the observations. And so you can do the same thing for the velocity. And then, of course, you can plug one into the other, and you end up with just a function of the positions at time, or at observation tau, tau plus 1, and tau plus 2. And so one thing to note is that this will allow you to work with data that's irregular in time, because h sub tau plus 1 does not necessarily have to be the same as h sub tau. So boom, we plug all that in and we get this crazy equation. So one thing to just note here is that the position at time tau plus 2 is just a function of the two previous positions. And then we have the motility in the error term. So it's going to show up in the variance of that error term. And then the motility and the potential surface are also included in that mean drift term. So to estimate the motility and the potential surfaces, we use, for this example, we use a spline expansion. So just degree 0, meaning it's just a piecewise constant function over the space, over the two-dimensional space. And so just looking at the picture here, if you zoom in, this is what it would look like. You just have this grid, and then you have different values of the surface in each grid cell. And of course, they look all really similar to each other. And that's because when we fit these and actually estimate these surfaces, we have to penalize the roughness of the surface. We don't want it to be really rough and jagged. We want it to look like a smooth surface. And so we estimate smoothness parameters using a holdout set. And to estimate that motility and potential, those spline surfaces, we use a Rummel-Seil approximation first to estimate the motility surface, because if you remember, the motility surface is actually in that variance. So we can estimate it from that, and then we go back and use that to estimate the potential surface from that mean drift. So it's a multi-step process, but it's very fast. So it's about 20 minutes for the full data, which is a decent amount of data. And this is what it ends up looking like. So this is the log of the motility surface. And so you see 4 different surfaces. The first, and letter A, is the full data. So that's fit with the entire data set. So we call that the closest thing we have to the true motility surface, so the true log motility surface. And then we compare each of the 3 subsamples to that. So you can see the 10-second Larry has a smaller mean squared error, where we're saying mean squared error is just summing over all the grid cells, the mean squared error compared to the full data motility surface. So it's smaller for the Larry than for the every 5 seconds. And you'll remember that these have the same amount of data. So you're directly comparing these sampling designs. And then similarly, we look at the potential surfaces. So I just kind of plotted them in 3D because they're cool. So the full data is in the top left. And in this case, you can actually see how these change when you do have less data. You do lose information about these surfaces. However, we can still look to see which one is better. So the 10-second Larry had the mean squared difference between that surface, the gradient of that surface, and the gradient of the true surface. So we use gradient because these are unidentifiable up to an additive constant. So we're comparing the gradients of these surfaces. And that mean squared distance is smallest for the 10-second Larry, even smaller than every 5 seconds and every 3 seconds. So that's meaningful because every 3 seconds actually has more data. So you actually were able to estimate the surface more closely than a sample that had more data. And of course, we could do this many times because the Larry subsample has that random component. So you can get a bunch of different Larry subsamples. And so we saw a similar thing in all of the cases that we looked at, where the potential surface mean squared distance was always smaller for Larry than it was for that one every 5-second subsample. And similarly for the motility surface, that mean squared error was always smaller for the Larry 10-second than for the every 5-second. So our results were the Larry sampling design did better than regular sampling overall. And in this paper, we looked at a simulation study as well. And we also looked at an additional data example with Guppy data, which also both of those kind of supported our conclusion. And in the simulated data, we also found that it could even be better for estimating missing data. But the caveat to that is just that it was better because it estimated the parameters better. So it seemed like just the fact that it did such a better job at estimating the parameters meant that it ended up working better for estimating missing data, even though you have bigger time gaps. So our conclusion is, regular sampling may not always be the best choice. We're not saying that Larry sampling is the answer to everything, but it is something that we should be looking into more. And so if you'd like to know more, you can check out our paper. And thank you so much.